Hello there, I'm Liz McDaniel. In this video, I'm going to talk about glycolysis and its reverse reaction, gluconeogenesis, as well as the enzymes involved in these processes. I will also discuss one specific enzyme involved in further detail, which is called aldolase, including its isoforms and genetic expression. This is for Chemistry 530, Concepts in Biochemistry with Dr. Kenneth Capps at Grand Canyon University. And just to give a brief overview before we get started, I'm going to, of course, provide some background on where and why glycolysis and gluconeogenesis occur and review the pathways for these processes. I'm going to talk about a special enzyme called aldolase and its isoforms and expression patterns, and I will also discuss how this enzyme is regulated and what transcription factors are involved in this regulation. As promised, here is some background information. Glycolysis comes from the Greek root words glykos, meaning sweet, and lysis, meaning to split. So this term literally means splitting sugar. It is the first step in cellular respiration, which of course results in the production of adenosine triphosphate, or ATP, which provides energy to the cell in every type of tissue. Glycolysis, in fact, does occur in every type of cell in humans. Gluconeogenesis does not, however. It occurs mainly in liver cells, but a few other cell types as well, and I will go over that in more detail when I talk about gluconeogenesis. The process of glycolysis will yield 2 molecules of ATP for each molecule of glucose, which enters the cell. Glycolysis also results in the production of pyruvate, which, depending on the availability of oxygen and the type of organism, will either enter into the mitochondria to take part in the citric acid cycle and complete cellular respiration, or will go on to a type of fermentation process, which in animals or mammals would be lactic acid fermentation, and in other organisms such as yeast, fermentation into ethanol and carbon dioxide. But I've gone too far. This presentation is just about glycolysis and its enzymes. Glycolysis occurs in just about every type of tissue, although this process is often associated with mitochondria, the powerhouses of the cell. This initial breaking down of sugar actually occurs in the cytosol. So once a glucose molecule enters the cell membrane, it is sent through a series of reactions, and the products of these reactions are what enter the mitochondria in order to produce even more energy-producing ATP. So let's look a little closer at this process in the next slide. You know, what's funny is that I remember learning about glycolysis in, I don't know, maybe high school biology, and I had the distinct impression that it was just one little reaction. And it wasn't until a few years later in college that I learned what a massive undertaking this pathway is. So as you can see in this image, glycolysis is a 10-step process, and it involves several enzymes which facilitate the breakdown of glucose. So these 10 steps can be divided into 2 phases. We have the preparatory phase, which consists of the first 5 reactions, and the payoff phase, which includes the last 5 reactions. This is where all of the energy comes from, these last 5 reactions. So let's review some organic chemistry before going through these steps. In rings, sugar rings like these, carbons have numbers. So if we look at this initial glucose molecule, each bend in this 6-carbon sugar is a carbon, except obviously for this little O, which is an oxygen. And we count them like this. We have carbon 1, 2, 3. This is 4, 5. And this top carbon is carbon 6. So the first step in this reaction is the phosphorylation of carbon 6, where a phosphate group is added onto this 6th carbon. This is facilitated by the hexokinase enzyme, and a single ATP molecule is put into that. And this yields a sugar called glucose 6-phosphate. The next step is isomerization, which is facilitated by phosphohexose isomerase or phosphoglucose isomerase. These are 2 different names for the same enzyme. And this moves that little carbonyl group to the 2nd carbon. So see now how we have a little pentagonal shape here. we have 1, 2, 3, 4, 5 sides. Now we have another phosphorylation on carbon number 1, which is catalyzed by phosphofructokinase 1, yielding fructose 1,6-biphosphate, and that's this guy right here. The next step is sort of a turning point for glycolysis, and it involves a special enzyme called fructose 1,6-biphosphate aldolase, which is generally referred to simply as aldolase. This is a step where that ring is broken, and this is an aldol condensation reaction, and two very similar smaller molecules are formed called glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Another enzyme called triose phosphate isomerase sort of interconverts these two, and they both wind up being pushed into the next phase as this one, glyceraldehyde 3-phosphate. And now we enter the next phase, which is the payoff phase of glycolysis. So now each G3P molecule is phosphorylated by an inorganic phosphate and forms 1,3-biphosphoglycerate right here. Note that an NADH is produced in this step for each of those 3 carbon sugars. And this is catalyzed by glyceraldehyde 3-phosphate dehydrogenase, that guy. In step 7, an ATP is produced from ATP for each of these small sugars as well. So these 1,3-biphosphoglycerate molecules are then converted into 3-phosphoglycerate by phosphoglycerate kinase, then into, which is that guy, then into 2-phosphopyruvates by an enzyme called phosphoglycerate mutase, and then finally into phosphoenolpyruvate by enolase, ultimately winding up as 2 pyruvate molecules and yielding 2 more ATP. Now, the gluconeogenesis pathway is effectively the exact opposite as glycolysis, and it uses pretty much the same enzymes as glycolysis in the reverse order, with a couple of exceptions at the initiation and in the final stages. So they're not exactly identical, but they do share 7 out of 10 enzyme reactions. So this process is important because in mammals, some tissues rely almost entirely on glucose for energy. So when glucose is low, there needs to be some way to synthesize it. For example, in the human brain, we require about 120 grams of glucose every day just to function. So when blood glucose levels are low, our bodies will utilize stored glycogen from the muscle and liver to create what is needed. This process takes place mainly in the liver, but it can also be accomplished in the renal cortex and in the epithelial cells of the small intestine. Interestingly, it does not take place in muscle cells, which is where most people think glucose is needed, but it doesn't actually occur in the muscle. So the start of our gluconeogenesis pathway begins with 3 or 4 carbon compounds such as lactate, pyruvate, glycerol, or even some amino acids. Gluconeogenesis begins with the conversion of pyruvate to phosphoenolpyruvate, or PEP. This is a 2-step reaction, which is irreversible, and it has a pretty big negative free energy change, making it an exergonic reaction. So this occurs when pyruvate, or another 3 to 4 carbon sugar, is transported into the mitochondria or is generated from alanine, and this is facilitated by an enzyme called pyruvate carboxylase. This forms an intermediary compound called oxaloacetate, which is then converted into phosphoenolpyruvate, which is there, by another enzyme called PEP carboxykinase, which is here. From there, the reactions in gluconeogenesis mirror those in glycolysis. So you can see these identical enzyme reactions moving upwards in gluconeogenesis with phosphoglycerate mutase, phosphoglycerate kinase, glyceraldehyde phosphate dehydrogenase, triose phosphate isomerase, and then aldolase, until it reaches the later stages in which fructose 1,6-biphosphate is converted into fructose 6-phosphate. Then we have another repeat enzyme, phosphohexose isomerase, catalyzing the next to last step. And finally, step 10, in which glucose 6-phosphate is converted to glucose by glucose 6-phosphatase rather than hexokinase. So these two are different. Now that you're familiar with these processes, I'm going to shift and discuss one of the enzymes involved, which is this one, aldolase. Now, the specific reaction that aldolase catalyzes is critical for both glycolysis and gluconeogenesis, as you saw in those previous two pathways. Fructose 1,6-biphosphate aldolase, or just aldolase, catalyzes that reaction shown here, where two 3-carbon sugars are joined to produce a 6-carbon fructose molecule. And as you can see by these little bidirectional arrows, this process goes both ways. And like most other enzyme reaction, the directionality of the process is dictated by the amount of either substance. So if there is a large concentration of these two, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, in the cytosol, the reaction will proceed to the right and catalyze fructose 1,6-biphosphate. But if there is a lot of fructose 1,6-biphosphate present, it's going to proceed with that aldol condensation and move in to the left. One interesting point here is that the standard free energy is negative when we're moving to the right, and meaning it gives off a lot of energy, and it is positive when it moves towards the left with the cleavage of this 6-carbon sugar. And if you've studied proteins or taken biochemistry, you probably know that there are generally many forms of enzymes called isoforms. Even in proteins that are technically the same, there can be some differences in specific amino acid sequences. And aldolase is no different. So there are actually two classes of aldolases, class 1 and class 2. Class 1 aldolases are found in animals and plants, and class 2 aldolases are found in fungi and bacteria and some protists. The difference between these two is that class 1 aldolases use a mechanism which forms something called a Schiff base intermediate, which is a little protonated area within the active site of the enzyme that delocalizes electrons to help facilitate those remaining enzymatic steps. Class 2 aldolases, on the other hand, are metalloenzymes, which utilize a zinc ion at their active site to facilitate that aldol condensation reaction to form triose phosphates in glycolysis, or to perform the opposite reaction in gluconeogenesis. There are actually 3 isoforms of class 1 aldolases. So we have isozyme A, which is found in muscles and red blood cells. Isozyme B is found in the liver, kidneys, and small intestines. And isozyme C can be found in smooth muscle tissue as well as in the brain and neuronal tissues. Class 1 aldolases are also nearly twice the size of class 2 aldolases. I'm going to shift now and talk a little bit more about this isozyme B, which is found in the liver, kidney, and small intestine, because this is the one that's responsible for a large portion of gluconeogenesis and glycolysis in the liver and kidneys. Just like every other enzyme and protein in the body, aldolase in our cells is regulated at the genetic level. The aldolase B enzyme is regulated at the transcriptional level by 2 important transcription factors, which are HNF1 and HNF3. HNF stands for hepatic nuclear factor. And I talked about how aldolase B is an essential enzyme involved in breaking down fructose in glycolysis and facilitating gluconeogenesis. It is mainly found in the liver, kidney, and small intestine. These are organs which serve to maintain metabolic balance. Regulation of aldolase B ensures that sugar balance is regulated and there won't be this accumulation of toxic byproducts. And it also allows cells to adjust enzyme production based on energy needs. So more aldolase means there's more enzymatic conversion of fructose and its 2,3-carbon products. HNF1 and HNF3 are transcription factors which are highly expressed in the liver. They bind to specific DNA sequences near target genes to regulate their transcription. So HNF1 plays a direct role. It binds to the aldolase B promoter region, which stimulates transcription. And this ensures that aldolase B is produced in liver cells, so it can manage fructose metabolism. HNF3 also binds to the aldolase B promoter, but it works a little bit differently. It actually helps to open up the chromatin structure, and this makes it easier for HNF1 and the general transcription machinery to access the gene and initiate transcription. HNF1 and HNF3 collaborate to ensure efficient gene activation. So while HNF1 directly triggers transcription, HNF3 prepares the table, so to speak. It creates this environment that supports the expression of aldolase B. So aldolase B expression is a very tightly regulated process. It's important for maintaining metabolic health. HNF1 is the primary activator, and HNF3 enhances those conditions needed for efficient transcription. And together they ensure that the body can effectively process glucose and fructose and maintain energy homeostasis. Mutations in these genes or in aldolase B can disrupt our metabolism and lead to diseases like hereditary fructose intolerance or diabetes. So understanding the regulation is really important in therapies such as treatment of diabetes or other metabolic disorders. And there are two other transcription factors which regulate aldolase B genes. So these are the CEBP and the DBP. Both are activators, meaning they turn on aldolase B expression when the body needs it. CEBP, or CCAT, enhancer binding protein, binds to a specific site called the CCAAT or cat box in the aldolase B promoter region. And this binding increases the transcription of the aldolase B gene, especially during liver development or when the liver just needs to adjust its metabolism. DBP stands for D-site binding protein, and it also activates aldolase B expression, but it is especially important for aligning gene expression with the body's circadian rhythm. DBP binds to the D-box sequence of the promoter, and this helps aldolase B fluctuate appropriately throughout the day in response to our eating and our fasting cycles. So these two together, CEBP and DBP, make sure that aldolase B is available when it's needed. CEBP provides a strong, stable expression during development and then also during metabolic shifts. And then DBP sort of fine-tunes the expression over the course of each day. Together, both transcription factors work to make sure that expression is consistent and timely, and they balance long-term development and daily metabolic demands. Okay, so I'm just going to summarize everything I've talked about because this was a lot of information. First up is glycolysis. This is the process where glucose is broken down into pyruvate. Along the way, the cell captures energy in the form of ATP and NADH. Glycolysis happens in the cytoplasm, not in the mitochondria, and is very carefully regulated at key steps by certain enzymes like hexokinase, phosphofructokinase 1, and pyruvate kinase. So moving on to gluconeogenesis. This is basically the body's way of making new glucose from other sources like lactate, amino acids, or pyruvate, and it mostly takes place in the liver and kidneys. Unlike glycolysis, gluconeogenesis consumes energy in the form of ATP, and it follows a similar path to glycolysis, but it just uses special enzymes at certain steps which are different, which include pyruvate carboxylase and fructose 1,6-biphosphatase. And finally, I talked about aldolase. This enzyme plays a critical role in both glycolysis and gluconeogenesis. It catalyzes the reversible splitting of fructose 1,6-biphosphate into 2 3-carbon molecules. There are different forms of aldolase found in various tissues, and it's important for maintaining metabolic flexibility. So overall, these processes are really important for energy management and adapting to different metabolic needs. And these are my references. Thank you so much.